Hello everyone, welcome to my biology classes. So this will be the fifth part from the biological classification chapter 2 from class 11. So here we will be discuss about virus, viroids, prions and lichens. Okay, this will be the last part from this chapter. Then next video I will be bringing on multiple choice question from this chapter. Okay, so let's start with virus, the topic virus. Okay, so let's see some characteristics of viruses. Okay, so they are non-cellular, means they do not have any cells. Okay, therefore they are non-cellular. All right, then the name, the virus, the name of the virus, that means that it is venom. Okay, it means venom or poisonous fluid. Okay, understood? And the name virus was given by Pasteur. Okay, so these points you have to keep in mind. Getting my point? So moving on to the next slide. So this is a picture of two different types of viruses. So what you can see that they are having simple structure. Okay, so they might have RNA or they might have DNA. Okay, and the covering, it will have a covering so that they can enclose this genetic materials, RNA and DNA. That covering is made up of proteins, that is called capsids. It is having outer layer. Okay, envelope. Okay, and in some viruses like bacteriophage, this is a picture of a bacteriophage virus. They are having head, the upper portion is head, then they are having neck, and they are having tail, and at the end they will have tail fiber. Getting my point? So this is the basic structure of a DNA. Just you have to keep in mind that they are having genetic material in the form of RNA also they can be having, DNA also they can have. Okay, and that genetic material is enclosed under a protein made cover that is called capsids. Getting my point? So moving on to the next slide. So here also we'll see some features of viruses, that viruses uses host machinery to multiply inside the host cell. They exist in a crystalline form outside the host cell. This means that whenever they are outside the host cell, okay, they will be in a crystalline form. Means they are somewhat inactive. Okay, when they get inside a host body, they will get activated. Okay, understanding? And if they get inside the host body, they will start activating, then they will start multiplying also. They will start multiplying. All right, then the next point is saying they are obligate parasite. What do you mean by obligate parasite? Means without host body they can't survive. They need a host body for their survival. Getting my point? And they need the host body for multiplication also. Getting my point? So they are called as obligate parasite and causes various diseases. In both plants and animals they causes viral diseases. Suppose in case of animals they cause common cold, AIDS, polio, mumps, measles, chicken pox, etc. Okay, so you have to know the names of the virus causing diseases in animals. Okay, for short questions. Next, in case of plants, they causes mosaic disease. They can cause disease in tobacco plants, cucumber plants, tomato plants, etc. So what are the symptoms you can see in the viral disease of a plant? That is, you can see curling of leaf. The leaf used to curl up. Okay, you can see yellowing of the veins. Do you know what is veins? In the leaves, the patterns, these patterns are called as veins. So these veins used to get colored as yellow color. Okay, so yellowing of veins. There might be stunted growth of the plant also. Okay, so there are many symptoms which we can see when there is a viral disease in the case of plants. Getting my point? So moving on to the next slide. So these are some diseases, viral diseases, which you can see in the plant. This is the curling of leaf. The leaf used to curl up. The yellowing of leaf. Okay. Clear? So moving on to the next slide. So here Ivanovsky was a scientist who gave the name virus to the tobacco mosaic disease. Okay, so what he found out that in tobacco plant, there he find out one disease, and he named that disease as tobacco mosaic disease. Okay, which caused due to tobacco mosaic virus. TMV is tobacco, T for tobacco, M for mosaic, V for virus. Okay, and he has identified it as a viral disease in case of tobacco plant. And here this value is for the size of the tobacco mosaic virus. The length is 300 nanometers, and its diameter, that is breadth, is 18 nanometers. Okay, understanding? This is the size of the tobacco mosaic virus, which causes tobacco mosaic disease in tobacco plants. Getting my point? So this, suppose if there is a infected tobacco plant and nearby there will be different plants, and if a tobacco plant is infected, then the rest of the plant will also get infected. Alright. And see this point. Infected tobacco can causes infection in healthy plants nearby present. Okay. So and here the fluid which has been extracted from the diseased tobacco plant, that fluid is called Contagium vivum fluidum. Getting? It is called Contagium vivum fluidum. The fluid which you extract from the disease, the affected tobacco plant. Okay, the fluid extracted from a diseased plant of tobacco. Okay, that is called Contagium vivum fluidum. All right. Okay, moving on to the next point. Stanley, he was also a scientist. He crystallized TMV, that is tobacco mosaic virus, for the first time. Means what he did, he extracted the tobacco mosaic virus from the tobacco plant which was infected, and he crystallized it. Okay, so he was the first one. Stanley, he was the scientist who first crystallized tobacco mosaic virus for the first time. Clear? So moving on to the next slide. This viruses are acellular, which we have already discussed, means they do not have any kind of cells. Okay, they contain nucleic acid core, means they are having either DNA or RNA. Okay, it might have DNA or it might have RNA, which is surrounded by a proteinaceous core that is called capsid. We have already discussed about it. This is only the theory part which you have to know. You can write down in your notes also. Okay, getting? And the viruses that mainly infect the plants. Okay, they are generally having single-stranded RNA. Okay, the virus which infects the plants, they will have single-stranded RNA. Clear? So moving on to the next slide. So here in this picture, we can see the structure of a tobacco mosaic virus. So we have already discussed that the virus that infects this plant will have single-stranded RNA. So here it is having single-stranded RNA, which is covered with a proteinaceous coat that is called your capsids. Understanding? Okay, so this is the cause, this is the result of the tobacco mosaic virus in the case of tobacco plants. Clear? So moving on to the next slide. So this is the Stanley. Remember he first crystallized tobacco mosaic virus. Okay, so this one he has found out. Okay, this is tobacco mosaic virus in the crystal form. Clear? Moving on to the next slide. So the another type of viruses is this bacteriophage. Okay, so bacteriophage are the type of viruses that infect the bacteria. They are viruses. Okay, bacteriophages are the viruses that infect the bacteria, and they are having double-stranded DNA. They will have double-stranded DNA. So just compare with tobacco mosaic virus. Tobacco mosaic virus is the one that infects the plants and that is having single-stranded RNA. Okay, and bacteriophage is a type of viruses that will affect the bacteria and they will have double-stranded DNA, and this is the structure of a bacteriophage. They are having outer coating as capsids, proteinaceous coating as capsids. They are having nucleic acid in the form of double-stranded DNA. They will have this is called head, we have already discussed. Head, collar, or you can say neck. This is the tail part. This is the tail fiber. Long tail fiber, here it is written. Clear? You have to know the structure of this virus. Okay, very important. Okay, moving on to the next slide. So they are having protein coat, which we have already discussed about capsids, proteinaceous coats they are having, which are, this capsids are made up of small units called the capsomeres. So small unit they are made up of. They are called capsomeres. Hold together they will form the capsids, the outer proteinaceous layer of the virus. Clear? Moving on to the next slide. So our virus part is completed. Now we'll start with viroids. Okay, what are viroids? So let's see its characteristics. They are smallest infectious agent found. Means they are much smaller than the virus. The smallest it is saying, na? Okay. The next point, they consist of nucleic acid only, but do not have any protein coat. Means the genetic material is freely floating. There is no outer coverage. Understanding? Okay. Diner was a scientist who discovered viroids as a causative agents of potato spindle tuber disease. So, this disease in potato is caused by whom? Viroids. And who discovered? Diner. He was a scientist who first discovered that potato is having a disease that is called potato spindle tuber disease, which caused by viroids. That was a free RNA because it does not have any protein coat. The free RNA only used to infect the potato. Okay, getting my point? Potato spindle tuber disease is caused by viroids. Okay, so let us see some pictures of potato spindle tuber disease. So this is the symptoms. Okay, this is the symptoms of that disease. Clear? So moving on to the next slide. Next topic is prions. Viroids are over, virus is over. Now coming to prions. What are prions? So let's see some characteristics of it. They contain abnormally folded proteins and have a size similar to viruses. So what is it? It is having abnormally folded proteins. Just talking about proteins that are abnormally folded like this. Okay, and the size is similar to that of the viruses. The size is similar, okay, to that of the viruses. They can change the shape of the normal protein by transmitting their misfold proteins. Means what it is saying that they can change the shape of the normal protein. Suppose in our body we have a normal protein, so they enter and change our normal protein to this abnormally folded protein. Okay, misfolded protein, or you can say abnormally folded protein, same. Okay, and they causes what they cause? They causes neurodegenerative disease. They causes some neurodegenerative disease. So let me, okay, sorry, wait, let me put myself up so that you can see clearly. Okay, they causes neurodegenerative disease. Example, bovine Spongiform encephalopathy. In what? In cattle. Bovine spongiform encephalopathy. This is a disease caused in cattle. And C.R. Jacob disease in humans. These are brain diseases. Okay. So these are caused by prions. You have to know the name. Okay. Clear? So moving on to the next slide. So next topic is your lichens. Will be the last topic. So what are lichens? So lichens are nothing but they are a symbiotic or mutually benefited association of one is algae and one is fungi. So there is a mutual symbiotic association with the algae and fungi. Suppose here I am writing one side algae and one side fungi. They are having a mutual relationship. Mutual relationship means both are getting benefited. They are staying together. Both are getting benefited from each other. Okay. So here in this case, the algae are known as the phycobionts. Phycobionts are the algae and mycobionts are the fungi. Clear? So the algae is autotrophic, means they make their own food with the help of photosynthesis. So they are making their own food. They are making food, okay, through photosynthesis and provides food. So they are making food, so they will provide food to the fungi. Getting my point? And the fungi, in replacement, they will protect and give shelter to the algae. So in place of the food, they are also giving shelter and protection to the algae. Getting my point? What is lichens? Okay. So next is lichens do not grow in polluted areas. Means wherever there is a pollution free area, you can see lichens, and whenever there is a polluted area, you can't see lichens. Okay. So they are a good pollution indicator. Since they are not present in the polluted area, so they are acted as a good pollution indicators. Okay. Clear? So understood what is phycobiont, what is mycobiont. Okay, so how they are symbiotically associated with each other. The algae is provided, he is doing photosynthesis. Since they are autotrophs, they are making food, they are giving food to the fungi, and in place what they are doing, fungi is giving them shelter and protection to the algae. Getting my point? So let us see some pictures of algae in the bark of the tree. You can see not algae, sorry, lichens. These are the lichens. This we can see only in the non-polluted areas. Okay, in the polluted areas this won't be there. Clear? So this is the end of this chapter. Hopefully you understood the chapter well. So next video I will bring some multiple choice question for your practice for your NEET or other entrances. So thank you for your patient hearing.